Okay, hello everyone. Welcome to the Neuroscience division. So we have 4 presentations in this division today. And at the end of each presentation, we will take questions from the judges and then the audience. And you may ask your questions through the chat or use the Zoom raise hand feature and then wait to be called on. And then before we begin the presentation, I would love it if the judges could briefly introduce themselves and just mention their research field.
Hi everyone, my name is Chen Liu. I'm currently a fifth-year PhD student in Dr. Michael Scollins' lab at Baylor University. Our research focuses on studying sleep and cognition, specifically in college students.
My name is Haley. I am a first-year grad student in Dr. Vichaya's psychoneuroimmunology lab. So we do a lot on how the brain and the immune system interact.
Okay, wonderful. So we're gonna just get right into it. So our first presentation is from Savannah from Baylor University. So you may share your screen and begin whenever you're ready.
Okay, can everyone see my screen? Okay, perfect. Okay. So, hello everyone. My name is Savannah Singer. I'm going to be presenting my research on the phenotypic behaviors of Fragile X syndrome in FMR1 mice on this background strain. And so, there we go. So the Fragile X syndrome is the most common form of intellectual disability, and it is comorbid with other conditions such as autism spectrum disorder, as well as ADHD. The symptoms can include hyperactivity, intellectual disabilities, as well as increase in certain behaviors, such as anxiety levels, repetitive behavior, as well as impulsivity. Fragile X syndrome is caused by the CGG trinucleotide expansion, and what it does is it hypermethylates the promoter region, and that causes the lack of the Fragile X mental protein expression. And you can see here on the picture at the bottom, the Fragile X chromosome, you can see at the bottom, it's definitely lacking in that protein expression. So the FMR1 mouse model, the knockout mice, they are similar to individuals with Fragile X syndrome as behavioral similarities, such as they have an increase in anxiety behavior and repetitive behavior, as well as some physical similarities, such as enlarged testes in the males, as well as the morphology of dendritic spines. The knockout mice and the individuals with FXS have immature dendritic spines. So you can see here in the picture, there are some examples of immature spines. So the purpose of my research was to further validate the behavioral symptoms of the FMR1 mouse model to provide further insight on those with Fragile X syndrome. And we wanted to expand the knowledge of Fragile X syndrome in the females because we did find that there was a large lack of research for the FMR1 wild type and heterozygous female mice on the C57BL6 background strain. The animals we used in this study were the FMR1 mice on the C57BL6 background strain. We used 73 mice total, 17 male wild type, 17 male knockout, 22 female heterozygous, and 17 female wild type. So we also did a timeline of the test. We started from least invasive to most invasive, starting with open field for week 1 and ending with free pulse inhibition. So starting with open field, we placed the mouse in the center of the chamber and their movement was evaluated for 30 minutes. And what it does was it evaluates for activity levels, which is the total time of movement within the chamber, as well as repetitive behavior, which is the amount of clockwise rotation the mouse performs, as well as anxiety-like behavior, which is the time and distance spent in the center of the chamber versus the outside of the chamber. So what we found in open field was an increase in repetitive activity for the males, but not the females. So we found that there was an increase in vertical episode count, as well as clockwise revolution in the knockout male mice compared to the wild type. We also found a decrease in anxiety levels in both the males and females. We found that the knockout males and the heterozygous females spent more time in the center of the chamber versus the surround. Our next test was the elevated plus maze. We placed the mouse in the center of the maze, and their movement was evaluated for 10 minutes. This evaluates for anxiety-like behavior as well as activity levels, and that is demonstrated by the frequency of head dips as well as the duration of time spent in each arm. So the open arm versus the closed arms. We did find that there was a decrease in anxiety levels, but just for males. So knockout males spent more time in the open arm duration, and they also had a more frequency of head dips compared to the wild type. We also did a marble bearing test in which the mouse is placed in the cage for 30 minutes. It does evaluate for repetitive behavior based on how many of the marbles were buried to 50%, 75%, 100%, and totally buried. We did not find any significant results. We did find that there was potentially some repetitive behavior in the females, and that there was an increase in the heterozygous females having 75% marbles buried. However, it was not significant. Next, we have tail suspension, in which the mouse is suspended by its tail and their movement was evaluated for 6 minutes. And it evaluates for depressive-like behavior by the time spent in which they are immobile. We did not find any significant differences or things like that in this test. Next, we have delayed fear conditioning, in which it was 3 separate trials over a span of 2 days. So day 1, we did trial 1, in which the mouse received the sound, and that is the conditioned stimulus, and then the shock, which was the unconditioned stimulus. And then on day 2, we had trial 2, in which the testing remained the same and their freezing was measured. And then on that same day, an hour later, on trial 3, we changed their environment, such as how it looked, the smell of the environment, and then we took away the shock and we produced the sound and we measured their freezing. This evaluates learning and memory deficits by the amount of time the mouse was frozen. And we did not find any significant results for this test. Next, we have pre-pulse inhibition, in which the mouse is placed in the chamber. And this had a span of about 2 weeks. And what it does is it evaluates for sensory motor gating abilities by how startled the mouse is based on different levels of sound. We did find that there was an increase in sensory motor gating ability in the lower decibels, especially for the males, as you've seen in graph D. However, as we got to higher decibel levels, there was no significant results. Overall, for anxiety levels, we did find that there was an overall decrease in anxiety levels for both the FMR1 male knockout mice as well as the FMR1 female heterozygous mice. And we did find that that was consistent with other studies of the same background strain. We also found that there was an increase in sensory motor gating ability for the FMR1 male knockout mice, and that was also consistent with other studies. We did not find any significant sensory motor gating abilities increases or decreases for the female mice. We also found that there was an increase in repetitive activity in the FMR1 male mice, and that was also consistent with other studies. However, we did not find any significant results in that area for female mice. We did not find any significant results for depressive behavior. However, in other studies, they did find an increase in depressive behavior just for males. However, there's not enough studies that did female mice on their depressive behavior, so we could not conclude anything. And then we have learning and memory. We did not find any significant results for learning and memory. However, in other studies, there was an increase in learning and memory deficits in both males and females. So there are subtle differences in the FMR1 female mice that could cause problems in research, and that we should have tests and where we can test specifically just on female mice and only focus on female mice to maybe catch those subtle differences. There are also some behavioral findings that may be dependent on environment. So the sensory motor gating ability, that could be based on genetic background, And also the anxiety behaviors, we tested for exploratory environment, kind of like anxiety. However, for Fragile X syndrome, the anxiety that is displayed there is more social and novel environment kind of anxiety. And so that could be not quite match up with the FMR1 mouse and the Fragile X syndrome behavioral phenotypes. And so there are differences between the FMR1 mouse model we tested and the Fragile X syndrome phenotype, such as we did find a decrease in anxiety levels, but for Fragile X syndrome, there's mostly an increase in anxiety levels. So I would like to thank you all for your attention, and I would like to thank the conference for allowing me to speak.
Okay, perfect. Thank you so much. So now I'm going to hand it off to the judges. If y'all have any questions you'd like to ask Savannah.
I do have a question. So I know that at the beginning of the study, you were wanting to look at the female mice, but it looks like most of the results were from just the male mice. Do you guys have any idea as to why, any theories behind why the females were less responsive than the males?
Yeah, so personally, I think that a lot of female, so in the behavior side, in the Fragile X syndrome in humans and individuals, most of the time females do display a lot more subtle differences, such as in autism spectrum disorder. Females are less diagnosed than males because they show different symptoms, as well as in ADHD. Females aren't as active as in males with ADHD. Their ADHD is a little bit different. And so it can be harder to diagnose females in the human aspect. And so for the mice, it's a lot harder to kind of find those subtle differences because there may be some differences between the symptoms of Fragile X symptoms in males versus like the female mice. And so that can be hard to find. And so for further research, if we just focus on the females and then not even do the males, we could perhaps find some more subtle differences and then we could emphasize on those. However, there's not enough research on the females because the male mice show the symptoms a lot more, and their symptoms are more exaggerated than the females. So that's why the females are less researched. So.
Thank you.
Great presentation. I really enjoyed it. I have a question. I mainly do human research, so I'm not very familiar with animal models, but I'm curious. So how did you decide your sample size? I see that you have 17 in most groups and 22 in one of the groups. So how did you decide to use 17 and 22?
So it's all dependent on the breeding. And so what happens is that we have a veterinarian in the BSB, and we tell them, oh, this is how many mice we need. And so we originally had more mice than this. However, the problem with mice is that sometimes they die. And so sometimes they die because it could be because of stress or different things like that. And so not every mice make it. And so we want to keep as much of our sample size as we can. And so that's why, there's 22 female heterozygous because they didn't die as often as the knockout males or the wild type females. I'm unsure why. That could be, something that we could look into. But most of the time, either mice die, or another thing could be that they either escape one of the tests. So if they escape one of the tests, like for elevated plus maze, if they fall off the maze, then we can't include them in the study. And so that is another potential block in having mouse models is that sometimes, if a mouse fails a test, then you can't really incorporate that into research because they didn't go through all the testing. And so, yeah, so that's why the numbers are a little mismatched because, yeah.
Yeah, that's totally understandable. I asked this because I see on one of the figures where there's no significant difference. I see there is a trend. So I feel like if you have more mice, maybe you will see a significant difference.
Yeah, I totally agree with that statement. Especially if we could, do another study, with more mice exactly the same way to kind of see if that trend continues and then maybe increase our sample size. Yeah, I totally agree with you.
Okay, y'all, thank you so much for those questions and your responses, Savannah. So we need to move on to the next presentation, but that was amazing. Thank you. So now our next presentation is by Dawn from Baylor University. So before we begin, judges, do you need any more time to finish writing feedback? Okay, perfect. So Dawn, whenever you're ready, you can share your screen and begin. Dawn, I think you're muted. You're so good.
I absolutely forgot, but thank you, Ruhi. And then, so hi everyone, my name is Dawn, and today I'm going to present on a recent research project that my lab has conducted. And this talk is going to be about a comparison of different maternal potentiation methodologies on C57 mice pups. And so with that said, I'm going to jump right in. And this is a brief outline of my talk. And so first, I will give an introduction on the term ultrasonic vocalizations and maternal potentiations, as those are the two terms that will be frequently used throughout this presentation. And then next, I also go over some of the literature review that has been done on maternal potentiation. And then after that, I will discuss the specific methodologies that our lab used to study maternal potentiation, and then the results that we got from that, as well as some conclusion that we have from our results, and then the future directions that we plan on taking to improve our results. And so first, I want to discuss ultrasonic vocalizations. And so these are isolation induced, meaning that they can be heard when the mice pup is isolated from its home cage at about 8 to 15 days of age. And these take place at the frequency of 30 to 90 kilohertz. And so because of that, these sounds cannot be heard by our normal ear, but we would have to use a special software to analyze this data. And so as you can see right here on the right, this is a screenshot of one of the USV projects that our lab used, that I was working on to analyze the data. And this is just one of the common pattern of USVs in mice pups. And so ultrasonic vocalizations is an effective study tool for neurodevelopmental disorders, early life impact, and more. And so in our lab specifically, we use a lot of USVs to study autism spectrum disorder, as this is as the disorder often involves communication deficits. And so USV is a great tool for that. And then next, I will want to go over the term maternal potentiation. And so this is a USV recording structure that has been previously established and more commonly conducted on rat pups. And so what we do is that basically there are two isolations in total. And so in the first isolation, the pup will be isolated from its home cage for a certain amount of time. And then after that, it will be returned to the home cage for reunion for a designated amount of time again. And then once that time passed, it will be re-isolated into the isolation box again. And the second isolation is actually designed to increase the USVs. And maternal potentiation is a frequently used model to study early life social behaviors, as well as relationships between the pup and the dam and its litter mates. And so next, I'm going to discuss some of the literature review that we have found on maternal potentiation, and kind of like what I touched on before. This model is well established in rat. And so as you can see right here in the figure on the left, there is a significantly higher amount of calls in the second isolation, ISO2, as indicated right here in rat models. And so the difference is clear in rat models, and the result is robust in rats. However, in mice models, the researchers have modified the protocol from that of rats, and also they have found that the greatest effect is seen in reunion when the mice is allowed to be with both its dam and the litter mate. And so you can see right here on the figure on the right, the greatest effect is seen when a modified protocol is used and the mice is allowed to be reunited with both the dam and the litter mate. And so the number of call is significantly higher the second time of isolation. And this is just overall also the goal of our study, is to see this effect in mice pups. However, there are also some limitations that we've seen that come along with maternal potentiation studies. And so there are still a scarce amount of MP studies on mice, as a lot of them also have been done on rats, but not a lot of mice. And so even with these scarce studies, we still found a lack of clear and unifying methodologies. And so this includes different isolation times, different reunion times, different isolation method, different reunion methods, and all of that. And so because of these various protocols, a lot of these studies have mixed results, which makes it hard to replicate these findings. And so with that being said, our goal for this studies is to experiment various protocols to find one or even multiple one that would produce the best result and the best effect for maternal potentiation on mice pups. So with that being said, I'm going to jump right into the methodology that we use to study project. And so in the first protocol, we allow all the animals to stay in their home cage. We'll first isolate a pup for 5 minutes and then record the USVs. And then we'll return the pup to its home cage and record the time of maternal retrieval. And then after 5 minutes of reunion, we'll re-isolate the pup again for 5 extra minutes and record the USVs. And then in protocol 2, this was actually similar to protocol 1, but this time we only isolate the pup and record USVs for 2 minutes instead of 5. And this was because we found that pups tend to emit a significant amount of calls during the 5 minutes time frame, possibly due to the stress and the cold temperature in the isolation box. So we found that it was most efficient to only isolate them for 2 minutes instead of 5. And the result that we found was still consistent even when there was a time difference. Then lastly, in the last protocol, this time we only utilized one female, so no males at all per breeder cage. And this was so that we could eliminate any complications that might have arisen with having multiple litters in a cage. And the steps that we follow were exactly the same as protocol 1, but this time we only used 2, 5, and 2 minute protocol as in protocol 2. And so the result that we got from this was that in protocol 1, there was not actually a difference in the number of calls before and after reunion, and there was in fact even a slight decrease in the number of calls in both the males and the females after reunion. In protocol 2, once again, there was a slight downward trend in the number of calls in males, but the number of calls in females stayed the same. So this is not the result that we were hoping to get as seen in the previous MP studies. And then in the last protocol, the number of calls in C57 males stay about the same again, but interestingly, in C57 females, the number of calls actually decreased significantly. So that was something that we also were not hoping to get, but it was interesting to see the result. And so the conclusions that we got from this was that our findings, unfortunately, did not support the protocol of maternal potentiation with the methods that we use. And this was because of going back to the idea of having scarce resource on maternal potentiation, and these studies were hard to replicate in our lab, and then also just some other factors that could have been better controlled. And so with that being said, future studies should use different protocols and provide more details so that other lab can replicate these findings as well. However, our lab is also taking future directions to improve our studies, and this involves developing new protocols with different housing conditions to test the result again. And then about a new protocol that we're currently developing and testing is in protocol 4. We'll separate the male immediately after the female is pregnant, and we'll test only a max of 2 males and 2 females from each litter. And this was so that from our result, we thought that a lot of the mistakes that happened was because of the effect of paternal potentiation, since the male was allowed to be in the cage. And so by separating the male immediately after the female is pregnant, we'll be able to eliminate any other possible variables that might have been involved in the study and just bring a special focus on the idea of maternal potentiation. And so with that being said, I want to say thank you specifically to my lab for helping me collect and analyze this data, and specifically thank you to Dr. Lugo for helping me put together this presentation and helping me with any troubleshoot that might have come along the way. But thank you everyone for listening, and I hope that you enjoyed the talk.
Okay, great. Thank you, Dawn. Okay, so now we'll just go back into if the judges have any questions for Dawn.
Yes, I do. You answered one of them because I was going to ask if the males were still allowed in the cage during that maternal potentiation time. So did you guys notice if there was a difference in the way that the dam treated the males versus female pups? And is that something that could have contributed to the males reacting differently? Or was that pretty standard between both?
So are you talking about having multiple litters in a cage and then the difference in interaction between?
Yeah, so in either case, if they were multiple litters or just a single litter, did you notice that the mother responded differently to the male versus the female pups that she was like going to grab?
Oh, okay. I don't think that was something that was noticeable. I think the maternal retrieval time was actually pretty similar among all of them, but there was just some difference in the way that they interact, in the way that like some moms are like, they have been moms before, and so versus first time moms, and so those were the differences that we saw in like the retrieval time. But I don't think there was a significance in the interactions between the males and the females.
Thank you. I think you did a really good job. Nice presentation. I don't have any questions. I wanted to ask, originally when you were presenting your conclusions, I wanted to ask what you could do to improve it, but then you went into protocol 4. So I don't have any questions.
Okay, great. Thank you again, Dawn. Thank you for our judges. Then students, anyone else who's a presenter, not a presenter, Joe, have any questions for Dawn?
Hi, I did want to know at what age you guys were doing the whole separation, how old the pups were whenever you separated them from the mother.
Oh, so our whole project focused on PD9 and PD12, but in this presentation specifically, we only use PD9. So those were 9 days old.
Okay. And then something that we've seen in our lab a lot of times, if our breeders get stressed, we actually, like they do, tend to like eat their pups. Did you guys have any trouble with that whenever you were separating them? Did it cause that kind of stress?
Yeah, I've heard of that happening in our discussion, and so I've heard a lot of mice pups. Like we expected a certain amount of mice pups, but we did not get that amount because they died during the process. Also, the dam or the dad or the mom or the dad just kind of ate the babies, which is kind of sad, and so that did affect our sample size too.
Thank you.
Okay, perfect time. Y'all are amazing. So I'm going to, do the judges have any more or need any more time writing your feedback, or are y'all good? Okay, wonderful. Y'all can take it away whenever you're ready.
Hello, I'm Dilmi Ranpitabendi, and I'm presenting alongside George Farag, Shreya Jajaria, Sona Subramanian, and Sabine Patel. Our teammate Daphne Seymour, who worked on this research with us, is sick and she won't be able to attend the symposium today, and we sincerely apologize. The topic of our presentation is factors influencing the alteration of gray matter texture features of the brain in medication overuse headaches. Medication overuse headaches, or MOH, occur as a result of consistent overuse of headache medications. Imaging studies were used to provide information about the neurological mechanisms of MOH, and these imaging processes consist of two steps. These two steps are gray matter segment and three-dimensional texture features mapping. In clinical studies, cerebral vermis of MOH patients texture parameters of increased contrast and entropy, decreased energy, and inverse difference moments were all targeted for analysis. In conclusion, the altered texture patterns in gray matter were elucidated to correlate in pain discrimination and modulation. Other studies have been conducted to identify the role of calcitonin gene-related peptide, which is also known as CGRP, and its potential contribution in pain processing in the context of MOH. Three-dimensional texture analysis and altered features of MR structural images over the entire brain were examined. Additionally, studies were conducted to test periaqueductal gray in migraine, regional volume changes over the whole brain stem, and several mechanisms involved in MOH.
The excessive dependence on prescribed medications to treat headaches has consistently brought more harm than good to human health. One such medical condition that affects those who consume regular, long-term medications to cure headaches is called medication overuse headache, which is also abbreviated as MOH. In an article on medication overuse headache, Benjamin Wakerly defines this term as a headache occurring on more than 15 days in a month to people with pre-existing primary headache and developing as a consequence of regular overuse of acute headache treatments. Medication overuse headache is common in general neurology clinics and can be difficult to manage. Most of these patients acquire a background with migraines, which has slowly transformed over months and years from the episodic to chronic form. And with this comes increased use of acute migraine treatment.
To visually depict our research process in an organized manner, we utilized a flow diagram to highlight the different phases of our meta-analysis review and find sufficient evidence to support a research question, which is elucidating the correlation between gray matter texture changes in the brain and calcitonin gene-related peptides, potential contribution to brain processing in medication overuse headache or MOH. In the initial stage of the research process, our group identified a total of 52 articles through a database search. After the screening and eligibility process, we narrowed down the reference articles to 28, according to criteria such as insufficient correlation to drug use, to gray matter changes, and findings not solely focused on the pain modulating pathways effects on the gray matter.
So the first study talked about periaqueductal gray. The periaqueductal gray is a descending pain modulatory center, and PAG dysfunction has been recognized in migraines. So the brain structural images were obtained from 32 normal controls, which will also be referred as NC, and 44 MOH patients on a 3.0 TMR system. PAG template was created based on the ICBM 152 gray matter template, and individual PAG segments were performed by applying the deformation field to the PAG template after structural image segments were performed. Gray level co-occurrence matrix, also referred to as GLCM, was performed to measure the texture parameters, including angular second moment, also referred to as ASM, contrast, correlation, inverse difference moment, also referred to as IDM, and entropy. This study revealed that the altered PAG texture contrast, which was presented in patients with MOH, were undetected by visual assessment, and might be associated with PAG dysfunction and considered as an auxiliary diagnostic. Additionally, it evaluated imaging biomarkers in MOH patients. The first image in the middle section shows a flowchart of the PAG texture contrast analysis performed, and the biomarkers used are the ASM, the contrast, the correlation, the IDM, and entropy.
Concerning the effects on the brainstem, a study was conducted to investigate the regional volume changes over the whole brainstem in the MOH patients using voxel-based morphometry in vivo. High-resolution three-dimensional structural images were obtained using a 3.0 Tesla magnetic resonance system from 36 MOH patients and 32 normal controls, also abbreviated as NCs, who were consecutively recruited from the International Headache Center, Chinese People Liberation Army General Hospital from March of 2013 to June of 2016. VBM was used to assess the brainstem structural alteration in the MOH patients, and voxel-wise correlation was performed to evaluate the relationship with the clinical characteristics. The brainstem region with increased volume were located in the ventral lateral periaqueductal gray and the ventral tegmental area, the bilateral substantia nigra, and the trigeminal root entry zone in MOH patients compared with normal controls. The headache visual scale score was positively related with the normal frontal ventromedial medulla in the MOH patients. In conclusion, the regional volume gain of brainstem could underlie the neuromechanism of impaired ascending and descending pathway in the MOH patients, and the left RVM volume alteration could imply the impaired tolerance of the nociceptive pain input and can be used to assess the headache disability of MOH patients. Referring to the second image within the middle section of our poster, the brainstem regions with the volume gain in MOH patients, MOH standing for medication overuse headache, patients compared with the NCs are the normal controls. The positive brain regions are located in the bilateral trigeminal REZ is the root entry zone, the bilateral SN refers to substantia nigra, the VTA, and the VTA stands for the ventral tegmental area, and the left ventrolateral PAG refers to the periaqueductal gray.
In a literature review, several mechanisms that have been linked to migraine headache and a potential role in MOH. For example, cortical spreading depression, which is also abbreviated as CSD, associated with migraine development is increased in frequency with prolonged use of topiramate or paracetamol. Results showed that increased CGRP levels in the blood have been linked to migraine, and elevated CGRP can be caused by prolonged sumatriptan exposure. Possible mechanisms that may be common to both migraine and medication overuse headache include increased endogenous facilitation of pain and or diminished endogenous pain inhibition. Neuroanatomical pathways mediating these effects were examined. Extensive research has demonstrated that frequent intake of pain medicine may serve to lower the threshold for experiencing pain and therefore reinforces the pathways that processes pain. Some medications that can cause headaches following frequent use are aspirin, acetaminophen, ibuprofen, naproxen, codeine, prescription pain relievers, caffeine-containing medications, and migraine drugs known as triptans. Pain medications have complex effects on the functioning of the brain. The bottom diagram in the middle section is a schematic diagram illustrating the interaction of descending projections involved in the inhibition and facilitation of pain. Placement of the rat's tail in hot water activates dorsal horn nociceptive neurons that project to the subnucleus reticularis dorsalis, which is also abbreviated as SRD. Descending projections from the SRD inhibit nociceptive transmission produced by the activation of dural afferents. In addition, noxious tail stimulation activates pain-facilitating neurons in the rostral ventral medial medulla, or RVM, mainly through indirect projections. Evidence indicates a shift in the balance between these two systems following chronic morphine exposure, such that descending facilitation dominates, making DNIC.
Although medication headache overuse is considered often to be a preventable medical condition, educating patients on the relationship between excessive use of acute medications and headache progression is an essential preventive measure. MOH patients seem to be mainly focused on the side effects that are related to the acute pain medications, which include gastrointestinal bleeding, kidney damage, and liver impairment. According to the article, A Critical Evaluation on MOH Current Treatments, patients are often surprised when they learn that the excessive use of acute pain drugs might increase headache frequency and thereby leading to the manifestation of MOH. The research concerning medication overuse headache is constantly moving forward in contemporary findings and has been successful in discovering mechanisms underlying headache progression and medication overuse. The article Medication Overuse Headache, a widely recognized entity amidst ongoing debate, states that whether MOH is a definitive distinct entity, a complication in the pathophysiology of primary headache disorders, or an epiphenomenon in the natural course of headache disorders is still up for debate. However, advanced technology and high-quality research will aid in resolving these numerous questions in hopes of helping clinicians make the right decisions and choices for patients who are suffering from MOH. Finally, the interconnectedness of the CGRP, pharmaceutical drugs, and pain modulation pathways are yet to be fully determined. However, this study offers possible insight into the relationship with one another. Thus, additional studies and analysis of literature should be conducted to consider the association of the aforementioned factors. Thank you very much, Dr. Klausmeier and Dr. Vichaya, for helping us throughout this process, and thank you to this platform for allowing us to speak and everyone here for listening.
Okay, perfect. Thank you so much. So now we're going to go to the judges again, if you'll have any questions for them.
So this study was done with rats, correct?
So only one of the aspects, which was to study the CGRP in blood, was done on rats. Just one of the aspects of it. Yeah.
Okay. So then what kind of behavior, was there any kind of like depressive symptoms reported from these patients as well as the migraines, or was it purely just the pain from the migraines?
It was purely the pain because they examined the pain modulatory system as well. So just the signals that were involved was just like the study was done mostly on like examining their pain. So yeah.
Thank you for your presentation. I think you mentioned that you conducted a pretty comprehensive literature review. And you may have already mentioned this, I may have missed that. Could you walk me through what kind of like keywords you used to conduct those review? What was the inclusion criteria for including a specific article?
Yes, definitely. So basically our main objective was how medication overuse headache is like, so if you increase your intake in medicines, instead of like decreasing your headache, it actually like makes it worse. And the thing that interested all of us, and which like made us like look for literature reviews on it, was like how medications that we take like on a daily basis can not only like it doesn't like it has like not only does it have like side like symptoms, but also like it can actually like make our pain worse. So we've all had like personal like experiences with like either our parents or like even our peers who like take medications on a daily basis because of their headache. So our main objective was to like see if these medications actually help in headache, because so we like looked up terms such as like medication, or like medication, does it worsen or like make your headaches better? So that was one of the main objectives. And after that, we found a lot of like research articles that was like associated with how there was a change in gray matter, and then the PAG, and then like all my other teammates, how they mentioned. So it was like a very extensive research about how all of these factors were influenced. Not just like the medication does not really like not only like increases or worsen your pain, but also like makes your gray matter and everything else like have a big impact on it. So yeah, our objective like led to a lot of like other subsidiary articles related to it.
Great, thank you.
Okay, perfect. So to all the presenters, all of y'all did an amazing job. So this kind of concludes the neuroscience division. So I'm gonna